Give me a second before we begin. All right, everybody ready?
Yeah.
All right, 3, 2. This is cell respiration. We're going to start in the cytoplasm of cells, where we have a 6-carbon molecule called a glucose. First thing that happens is we've got to spend some ATP by firing off the phosphate, converting this into ADP. The energy that we released converts this glucose into 2 3-carbon molecules called pGALs. Now, for the remainder of this process of glycolysis, we do a couple of things. First of all, each one of these molecules takes a hydrogen and attaches it to an NAD to form an NADH. It also attaches a phosphate to an ADP to form a TP, and does that a couple times each. And now we're at the end of glycolysis, and these molecules are no longer pGAL, they're now called pyruvic acids. From this point on, everything that happens to one of these happens to the other one, so I'm only going to show you one of them. Just know it happens with both of them. I'll move this stuff to the side here, and I'll just do this one right here. So this pyruvic acid is going to go into the mitochondria, and it's going to do the intermediate step before we go into the next process. And the intermediate step is just one quick step. Rip off one of the carbons as carbon dioxide and exhale it out of the cell. And while we do that, we're also going to attach a hydrogen to an NAD and form an NADH. And that's the end of the intermediate step. We are ready to do the citric acid cycle. Now this whole time, there's been a 4-carbon molecule inside of the mitochondrial matrix. That's the innermost section of the mitochondria. The 2-carbon molecule we ended with is going to bind to this 4-carbon molecule to temporarily create a 6-carbon molecule called citric acid, hence the name citric acid cycle. Over the next 8 steps, we're going to do 4 things. First thing is we're going to make NADH. In fact, we make 3 NADH for every cycle. We also will attach a hydrogen to an FAD to form FADH2, and we make 1 of those per cycle. We make another ATP for every cycle, and we got to get this molecule back to its original 4-carbon molecule. So we take 2 carbons off and eject them as carbon dioxide. And now we are done with the citric acid cycle. The last step uses all of the NADHs and FADHs, but I'm only going to show you one of those, so I'll just move the rest over here. Now this whole time, there's been a pile of hydrogens living on either side of the inner mitochondrial membrane. And they're always at equilibrium. We're going to mess with that. We're going to take the NADH and we're going to fire off the hydrogen so that it activates the proteins in the electron transport chain. First protein's activated, pull a hydrogen across. Next protein activated, pull another hydrogen across. Last protein activated, pull yet another hydrogen across. Now these hydrogens are not at equilibrium. They're going to want to get back across the membrane, and they will do so through ATP synthase. And every time they go through ATP synthase, it builds an ATP. Another one goes through, and so we build yet another ATP. 4 and 2, still not at equilibrium. So we're going to send another one through and build yet another ATP. 3 and 3, now we're at equilibrium. That part's done. The only problem is that this electron from the NADH is still sitting there. It's plugging up the electron transport chain, and we got to get rid of it. So we're going to grab oxygen that we inhale. It's going to be attracted to the hydrogen, and it's going to convert all of that into water that is exhaled out of the cell in some way. And that is the end of cell respiration. All right. Thank you.